Hello, everybody. My name is Jun Wan. I'm an assistant professor at the Department of Medical and Molecular Genetics, and I'm also directing the collaborative call for cancer bioinformatics C3B cohort. And today, I'm going to show you a demo of RNA-seq analysis. We will perform the demo or show you everything on our computer system, supercomputer system. First, I would like to thank Dr. Shen Liu from my lab to prepare all the materials for this demo. This is IUCAST cluster, the supercomputer system we are using to show our demo. After logging, and we are go to a folder on the C3B workshop. There are already 4 subfolders under this folder: FASTQ, outputs, results, scripts. We put the different files on the different subfolders. And on the FASTQ, we may see the sample FASTQ file here. And in this case, we only put the 2 samples, A and B here. And from this one, you can see each sample has 2 files indicating pair and RNA-seq analysis. And for the outputs, we just put all outputs file there, and we summarize the output as a final result. We will move them to the results. And the script, including all the scripts we are going to use to show demo today, which includes the major one is a star feature count from the alignment, from the trimming to alignment to the feature count. All the majority of pipelines will be shown in this SH file. And of course, and Shen also brought some R code to summarize all the results, including the D analysis, which will use HR to do differential analysis. This SH file, including all steps from tumor to star to feature count. And let's go through the steps one by one. And the first you have to see, and you can see we have to load all the module available from the supercomputer, including Java, Chimgalo, SAMtools, which is frequently used by bioinformaticians, and the star as the default aligner in our pipeline. And also, as I mentioned before, we use NGS utils to perform statistics on the response. And the first, you have to define the input variables, like what's the sample, and where the work folder, the director, something like that. And also, there are other applications we are going to use in this pipeline. The first one is the feature counts. Underline where the feature counts located on the supercomputer. And this is a number of running threads. And also, one important thing is we have to define the reference genome we are going to use. And there are several reference genomes GTF file we need. The first one is we need the GTF file for the gene annotation. We may use as a feature count, like we know where the race come from, which gene they come from. And also there is another annotation file, which is the same one as the same list version, but this will be used for the BAM status. We count how many lists can map the gene exon, gene intron, and junctions, and the intergenic promoter, 5' UTI, 3' UTI mitochondria, something like that. This index is for reference genome, and we have a different index file depending on the read length. And actually here we have for human genome, we have 3 files which corresponding to different read lengths 50, 75, and 100. The next step is you have to prepare output directory. like where the output file you are supposed to put on, and because some output file may be used for next step analysis like HR for differential expression analysis. After you're ready, next we are going to do the first step of RNA-seq analysis, trimming adapters. We use a trim getter to trim adapters if there are many adapter sequences in the FASTQ file. And also you have to mention whether your RNA-seq is a single end by default or paired end. You have to underline here. After trimming, then next step is you need to map to reference genomes. We use star, and first you need to set up the number of running threads, and you have to point to the index director where index file locates for the human genome, for the reference genome, and then you put the read files input here. And if it's a pair end, you need to put both read and file in here. And then you name the output folder and where they locate, they should put on those BAM files. And the last step also, you are going to use the same tools to index all the BAM files. After mapping, even you find some reads can be mapped to genome region, but still, maybe the mapping quality is not good, so it's better to filter out those reads that can be mapped to multiple genome locations or with a very low quality. Here, we use the MAPQ10 as a cutoff, which is kind of commonly used in many studies. So we use a BAM filter filter those low quality reads, and here is the cutoff MAPQ10. Of course, you may set as 20 or whatever you want to choose. Then we get the output file after same tools index. Then we have a file. So basically, you name the MAPQ10 filtered BAM file. As long as we filter low quality reads, then we have to summarize all alignment metrics into file. And we use the BAM utilities and do status analysis on those reads. We try to find how many total reads and how many reads can be mapped to the reference genome, how many of them have a high qualities. And then we may have an idea whether the sample is good or not in terms of mapping. Again, this is RNA-seq data, so it's not yet. Next main step is we need to count the number of reads in terms of gene or transcript or even exon. So here we use the feature count to try to find on each gene or each exon or each transcript how many reads can located on that region. And this file will be the summarized gene expression we will use for further analysis. After you finish the backbone for RNA-seq analysis, you may think about how to submit the job to IU supercomputer system to run the analysis. Before doing that, you have to first put all the samples in one file. Like in this file, you show the samples RNA-seq loaded FASTQ files, where they located, and what's the sample name, A, B, C, D, something like that. And also you have to write another script and show the comments you need to use and where the directory, what's the sample file, how many samples you are going to use, and what director and the species and the read length. As you can see, those are very important information and the memories you required. Then you can submit your data to the IU supercomputer system. you can see, and this is a matchup samples sh. This is the input files, and this is the photo we used, and that's the memories you ask for, and the hours or running times you ask for. When you get ready everything, just submit, then waiting for the result. After your job is done on the IU supercomputer system, you may find your results under the folder you named. Here, example, we named on the folder outputs. So for each sample has multiple files, including all the information, like a BAM file after the mapping, and the BAM file with the index, and the BAM counts with all the gene counts, and also BAM status showing the status on the gene body, and also log file. In order to merge all the files in one table, you have to write your own code to do that. Here we have two custom code in R. We merge BAM status together in one file and merge all feature counts together in one file, and we put those files in the results. So we have two output files here, red status in Excel file and the gene counts in text file, which will be used for next step analysis, differential gene expression analysis. After merging all future counts result into one file gene counts, we are going to start the differential expression analysis. We run differential analysis in the R, and here is the comments in the written in R. And first we need to point the input count file, and the meta index shows the column number for gene information count matrix from number 1 to 6. And also here we have 6 samples together, 2 conditions. So we use the column number for count data for samples in count matrix. And here is the gene ID, and the symbols will be used to exporting differential express genes. Now, here we set the word directory, and also we read all the counts, input counts, and also including samples. Then we have to, first we need to convert counts to D-list objects and put counts, gene information, group information together. Then next step, we need to define our design matrix based on the experiment design. Like for example, if we want to test for differential analysis expression between B and A, then we need to design to make 2 groups. And also we need to filter out genes with low risk counts in either of the conditions. Normally, if we said, okay, over 2 cores or 3 cores, and all 3 cores genes, they have a low expression, and then we maybe filter out. We don't want to do differential analysis on those ones. And then next step, we have to do the normalization, as we mentioned in the workflow. And the trimmed, here we use the trimmed mean of value normalization method, TMM, and to eliminate composition bias between libraries. And here, as I said, okay, we can do either MDS plot or principal component plot, PCA plot. And we have to, first we need to change the gene expression in terms of log scale with base 2 of count per million. And then we do PCA analysis in the package. And then we plot the PCA plot and output the PCA plot to the final result. If everything is fine, I mean, if we see, okay, the sample quality looks not weird from the PCA or MDS plot, then we can take all samples into differential analysis. First, we also have to estimate the gene-wise dispersion over all genes. So in other words, we are allowed for a possible abundance chain. And then we use the generalized linear model to fit the whole gene expression. And then we make the contrast, like we compare the groups of our interest, like we just simply compare group 1 versus 2, or maybe we use different comparison like pairwise, or we remove batch something, and then we conduct a likelihood ratio test for 2 group comparison in this example, like B versus A for tissue comparison. After we do everything, we can merge gene information, normalization count, into the final differential result, including fold change, including p-value and FDR. And then we output the result to somewhere in the Excel file. And of course, if you need it, and we can set the default cutoff, like normally FDR less than 0.05 and log fold change larger than 1, and then we output all the gene differential expressed genes. And we show you which one is down-regulated, which one is up-regulated. And this is the final file, D genes. Here are output from the differential express analysis. D genes, including all the genes with the information from differential analysis, and the MDS plot and or PCA plot. And in next lectures, I will go through all output files and show you how to read the files, how to understand the data. Thank you.